Click for IB Biology. Polysaccharides, the large polymers of monosaccharides, and here we're particularly going to focus on starch and cellulose, both of which we find in plants, and we'll also then take a look at glycogen, which is an animal polypeptide. Should you wish, there's a PDF in the description that you can download and annotate with your own notes, and later you can take the quiz which is down there as well to make sure that you have understood the content. So let's deal with starch first. Starch is an insoluble and unreactive polymer of glucose, and because of these properties, insoluble and unreactive, in plants, this makes it an excellent storage molecule. Insoluble, it doesn't affect osmosis. Unreactive, it doesn't unnecessarily get involved in chemical reactions. Starch is actually composed of two different types of molecule. One is called amylose and the other is called amylopectin. The simpler of the two is amylose, which is composed of a long chain of glucose molecules showing 1,4 glycosidic bonds, and it actually tends to form a spiral shape, as I'm trying to show in this diagram here. Amylopectin, on the other hand, about every 25 to 30 sugars, it does a 1,6 bond, and that's what creates these branching occasions like this. Amylose rather, if you have higher amylopectin to amylose ratio, then it becomes slightly more soluble. So plants can adjust the ratio of amylopectin to amylose to change the solubility of starch. Over here we have the animal storage molecule polymer of glucose, which is glycogen. Largely composed of 1 to 4 glycosidic bonds, as we've shown you already in videos. But then we have 1,6 bonds, every 12, so that's much more frequent than we had over in the amylopectin, and that's why the branching that you see here is more frequent, making the glycogen just that little bit more dense. And so animals store glucose as glycogen, which is insoluble and it's unreactive, as we've written here in fact, and this means that insoluble, it has no osmotic effect. Unreactive, does not unnecessarily get involved in chemistry. We can store, as humans, we can store about 1 kilogram of glycogen, 100 grams in the liver, but much more in our muscles, and that's a topic that we'll visit later when we're looking at muscle physiology. The third polymer of glucose is cellulose, and now we're back to plants. And the significance of this one is its role in the construction of the cell wall. So the cell wall is composed of cellulose, and you can see here the chains of glucose molecules with the alpha 1,4s, and then they're hydrogen bonding across, like so, to create this very high tensile strength structure. It tends to be linear, held together by hydrogen bonding, and very resistant to digestion unless you have cellulase enzyme. If you have cellulase enzyme, you can digest cellulose, but that's restricted largely to bacteria and to fungi and their roles in decomposition. The structure of cellulose as shown here is very fibrous and it's extremely permeable. So it doesn't really resist the movement of molecules. That's the job of the cell membrane. Further videos in the carbohydrate series are now being shown in the links, along with the opportunity to subscribe and a link back to our playlist.